1,000 individuals are found to have come down with MS. And in US alone, they spend approximately $28 billion in lost productivity as well as medical expenses. So what exactly happens in multiple sclerosis? Now, there are the T cells which are reacting against self-antigens that get activated. These are the CD4 positive T cells, which include the Th1 and the Th17 cells. And these cells produce certain cytokines, such as the gamma interferon and IL-17, as well as these autoreactive T cells can cause demyelination. And that leads to neuronal damage, and this results in paralysis. So we have used a mouse model of multiple sclerosis, which is known as experimental autoimmune encephalomyelitis, or EAE. So what we do in these is that we administer an antigen known as myelin oligodendrocyte glycoprotein, and in complete friends adjuvant, along with pertussis toxin, and when these mice are monitored daily for signs of paralysis. Now, into these mice, we administered these indoles one day prior to the administration of this antigen, the MOG antigen, and then we have kept treating these throughout a period of about a month. So when we found that, as you can see, the EAE mice showed signs of the disease, whereas there was complete decrease, or there was no disease seen following treatment with I3C or DIM, thereby showing that these indoles are quite effective in causing the disease does not develop in these mice. So we looked at the histopathological analysis of the spinal cord in these groups, when we found that in the EAE mice there is significant infiltration, as can be seen in the second panel, this infiltration was because of the mononuclear cells. Whereas following treatment with I3C and DIM, there was no such infiltration with mononuclear cells in the spinal cord of these mice. We also did late treatment, wherein we treated these animals at a later point in time after the disease was induced. And we found that these indoles were also efficacious of the infiltrating cells in the brain of these EAE mice. And as you can see, we stained them with the antibody